Okay, thank you all for your patience and apologies for that. There's never a Zoom webinar that doesn't start off with a little bit of a hiccup. So again, thank you for your patience. Good morning to everyone. I am very excited today to welcome you all to the 2023 Research Administrator Symposium. Just to let everybody know, we have a current count of about 106 participants. So that is a great number to start with. And hopefully we just continue to have some people continue to join while we're getting started, and also before our great keynote address that we have scheduled for today. So just to get started, if you are not logged in with your UNM or UNM HSE account, you may want to take a moment to do so. We've set up the breakout rooms based on UNM and at Salud addresses. So just a quick reminder there if you're not logged in. Again, we've been doing this for about 7 or 8 years now, and the last 3 have been held virtually. Then we started going virtually because of COVID, and we've just found that our attendance has been so much higher, and this symposium has been well attended virtually that we've decided to keep it virtually this year. So thank you all for joining us again. Planning and executing any conference or symposium is very difficult, whether it's virtual or in person. And it does take an enormous amount of time, flexibility, and a lot of learning. So I do want to give a special shout out and thank you to our planning team, our planning committee, that is composed of Lucas Reynolds, Monica Fischel, Grace Faustino, Teresa Hammett, Gloria Quintana, Sam Mascarenas, Marissa Lucero, Rayanne McKernan, Janine Pacheco, Maureen Torresini, Jessica Hermosillo, and Kate Charleston from HSC. So thank you to them for all of their hard work in making this a successful event. They've been diligently working and making time in their busy schedules for this and having weekly meetings since about June. So thank you very much to them. I know that was a long list of names, but I'm actually very happy that it was, because it had gotten pretty short in the previous year. So thank you to those who have recently joined us, as well as planning member committees. I do also want to thank our presenters and moderators for embracing this opportunity to develop the outstanding program that's being presented this year. I'm very proud and excited about our presentation lineup this year. I think it's one of the best ones yet, and it just continues to get better. And again, this is work that people take on to share the knowledge that they've been able to gain over the years with other research administrators and with new research administrators. So thank you very much to our presenters for taking on that extra work and doing this. And finally, I would like to thank each and all of you for making time in your busy schedules to join us today and tomorrow, and also for all the work that you do to make UNM's research mission successful. You get to hear some great talks, and it just shows that UNM, as the state flagship and R1 institution, is rightfully in its position. But before we get started, just some last reminders to keep in mind during the next 2 days, and kind of some housekeeping items. The workshops will be held, all be held in breakout rooms. Again, that is based upon your registration, and it should be auto-assigned. But if at any time you find yourself in the wrong breakout room, or you just want to change the session, feel free to go back to the main room, and someone will be there to be able to assign you to the correct or a different breakout session. At the end of each breakout session, we will return to the main room until the next session begins. I would ask, just please remember to mute yourself. If you'd like to ask questions of the speaker, we do have an opportunity. You can raise your hand, and the moderator will call on you to unmute and ask the question. And you may also ask questions in the chat, but the moderators may be going to raise hands before the chat. And so without further ado, I would like to take this moment to turn the event over to our Vice President for Research, Professor Ellen Fisher, for welcoming remarks and to introduce our keynote speaker. Dr. Fisher.
Thanks, Greg. And I will just add a quick welcome to the 2023 Research Administrators Symposium, and add my thank yous to the RAS Planning Committee, the Research Administrators Network, and also to all of our research administrators on all of our campuses. As you know, research administrators are absolutely vital to everything we do as an R1 institution, and we certainly could not succeed in any of our research endeavors without you. The work you do and the support you give to UNM researchers are absolutely critical in allowing us to reach our research goals as a university. Indeed, you guys really do make what we do, research, possible. You make it happen. And I'm really honored to be with you here today. Before I introduce our speaker, I thought I'd also just share with you some of our numbers. These are sort of the basic research metrics that we use to measure success in some ways. And I would say that I'm continuing to be astonished at how we have really stepped up on main and branch campuses. Our F&A expenditures in FY23 totaled more than $146 million, which is a 13.5% increase from FY22. And in addition, our F&A recovery for FY23 totaled more than $25.8 million. This was also a 13% increase from the previous year. Which is amazing. Double-digit percentage increases is really awesome. And the first 2 months of FY24 have also started off just as strong. Our research expenditures are up once again by more than 12.5%, and our F&A recovery is up more than 12.7% compared to the first 2 months of last year. So we're continuing on this upward trajectory. And again, a lot of that is in part due to the research administrators that helped to make it happen. And certainly getting proposals submitted and getting awards set up are really key to us being able to actually spend the money on the research that we do. One other kind of announcement is I hope that all of you will find ways to attend any of the conferences, lectures, workshops, and open houses that are planned for our second annual Research and Discovery Week, which is less than 6 weeks away. It is going to be held November 4th through 11th, and there's a lot of really awesome stuff that's happening. And we're also looking for ways to directly engage our research administrators in a variety of activities. And really, we're just interested in celebrating and highlighting the research that we do at UNM across all campuses, and that's what that week is all about. So with that, it is absolutely my pleasure to introduce today's keynote speaker, Professor Mubarak Hussain Syed, who many around campus know as the Fly Guy. And it's not because he flies airplanes. I'll tell you in a minute why that is, why he's called the Fly Guy. Dr. Syed grew up in a small village in the remote Himalayan region of Kashmir. Other than brain research, he is a staunch advocate for education science, diversity in science, and open access. Along those lines, he has founded an organization, JK Scientists, that seeks to reach and mentor students from disadvantaged backgrounds. In New Mexico, he has started an ambitious Pueblo Brain Science project that aims to train a diverse force of next-gen neuroscientists through training, mentoring, and outreach to the local communities and schools. We in the OVPR had the pleasure and opportunity to partner with and help Dr. Syed offer a new discovery-based course called NeuroCure, meant to engage and train undergraduate researchers in neurogenetics. Dr. Syed is the recipient of an NSF Career Award, was recently awarded a Sloan Fellow in Neuroscience, and also the Brain and Behavior Research Young Investigator Award, which was formerly known as NARSAD. I'm not sure what that was, but anyway, Brain and Behavior Research Young Investigator Award. I look forward to hearing and learning from Dr. Syed's work from his presentation today, which is entitled Tiny Brains, Big Impact, Using the power of fruit flies to understand brain development and disorders. Dr. Syed?
Thank you very much for this nice introduction and also for inviting me here to talk to you. It has been 4 years at UNM, and OVPR's office and people who work in OSP have been very helpful. Before I start my presentation, I would like to thank you all, because when I started faculty as a postdoc, you don't know much about budgeting and all this, how to submit a proposal. And the first proposal I submitted was, in fact, NSF Career, which I was lucky enough to get in the first go. Thank you all for your work, and keep us doing what we do best, is research and teaching. Thank you. Now I can share. I have made my presentation kind of more general, but also specific to what we do here, so that you all know what power lies in fruit fly brain. So do you see all the screen?
Yes, we do.
And am I audible? Thank you. So you see on the right hand, there are these small, tiny fruit flies. Each tube has one fruit fly, and over days and nights, you can track their activity if they are sleeping or not. So fruit flies indeed sleep. We will talk about that later. And in the green here are sleep neurons of a fly brain. Similarly, in our brain, we have sleep promoting neurons. So this is the adult fly brain. And we understand how these different kinds of neurons, which tell fly to sleep or to fly, to navigate, develop during development. And I'm called the fly guy because I work with fruit flies. So like Dr. Official said, I'm from Kashmir. This would be Kashmir. I don't know if you know the Himalayan range. It's on top, which is bordering Pakistan on this side, and on India will be on the other side. So I did all my education there, master's there. Then I moved to Bangalore, which is south of India. Then I did my PhD in Germany, postdoc in Oregon, and started my faculty position here at 2019. Before I go further, I would like to show my lab, who actually did the hard work. And this is UNM, and this is my office, which I am here now. All the undergrads, high school students, the PhD student, master student, undergrad. So this is how Fly Lab looks like. This is me in the office. Undergrad, research scientist. So he's the first PhD student who actually did lots of work for what I'm going to tell you today. And what we are interested in understanding is how does this billions of neurons, how do billions of neurons in our brain form during development? Our brain has more than 80 billion neurons, and there are more than other cell types, which are called the glial cells. And these neurons are almost equivalent, are more than the stars in this galaxy. It's amazing, astonishing. And all these neurons are of different kinds and different shape. For example, if you see, this is a cartoon of cortex. Cortex is the higher order brain region or center in humans. Starting from the deeper layer to the upper layer, there are different classes of neurons. Neuron will be the brain cell. There are different kinds, different shape, and they connect to different neurons to find circuits, to find the circuit where they should belong. And then at the end, we end up with a functional brain. So this is 1 month after gestation. 2 months, you see some brain cells will be called stem cells. They will divide, and over 9 months, we will have almost all of the neurogenesis, making formation of all these billions of neurons done, complete. And this months, initial months when we are in our mother's womb is amazing, is the main time when all the neurons are made. So what happens is these stem cells called neural stem cells over time will divide, and the ones which are born early will stay closer to the mother cell, which is called stem cell here. And the ones which are born later will migrate out to the outer cortex. So they end up in the outer layer of our cortex. So that means the same stem cell over time will generate different classes of neurons, older ones, mid, and younger ones. How does this happen? And this is what we are trying to understand in a small, more tractable brain. And if something goes wrong during these integrate timing during development, we end up with disordered brain. And majority of the neuropsychiatric disorders are developmental in origin, means something goes wrong during development when we are in our mother's womb. And now you are in your cortex, and in the cortex, if you see, are billions of neurons, trillions of connections, and lots of activity happening. And behind all this activity are circuits, which are composed of unique cell types. And these unique circuits and cell types are very important for anything we do during our day, waking up, having coffee, coming to our work or office, and then deciding what to do, and also later on for sleep. So as I mentioned, most of the neurogenesis, means formation of these billions of neurons, happens during development. before we are born. And when we are born, first 5 years, most of the neurons have found their position, then they mature, the circuits mature. And this is comparative to the mice one. If there is some either genetic defect or environmental also has a huge role, like in New Mexico, majority of the neurodevelopmental disorders are due to fetal alcohol syndrome, because parent or mother during gestation drinks a little bit of alcohol and the brain gets deformed because it affects development, and that brain defect or disorder is irreversible. It cannot be repaired. That's why development or studying development is so critical. And many of the autism spectrum disorders, neuropsychiatric disorders, like Dr. Fisher said, I got NARSAD, which is not public yet, but NARSAD stands for National Alliance for Research in Schizophrenia and Depression. So most of these disorders have developmental component. And I guess they believe that flies can be a good system to understand these disorders. So as I mentioned, there are different classes of neurons in this cortex, which if you see on the left side will be a cartoon or schematic of stem cell over time, keep dividing and generating distinct classes of neurons. How that happens is not properly understood. And that's what we study in our brain. We don't study human brain because of its complexity and also ethical issues. Mouse also too complex and also very hard to go to the single stem cell level, which you will appreciate in my talk, why flies are good. And this is the fly, it's a dot. But it's very useful, has been useful in understanding genetics and neuroscience and development. And also I'm fascinated by the insect world. Like many of you may be familiar with this long migration butterflies do from Mexico to Canada. This dung beetle rolls the dung towards the nest in opposite orientation. All these insects for navigation use celestial cues. So their brain is highly sophisticated. They integrate all the information, then make decisions like we do. For example, this ant in the desert, when they forage for food, it takes some time to get the food, but once they reach the food, they calculate the shortest possible distance. And all this is because they have integrated all the information like we do from environment and also direction from the solar and also solar azimuth. They integrate into the brain center, which is called central complex. So we will be talking a lot about this insect brain region called central complex. Please interrupt me if I'm going too fast, if it is too much. I tried to make it simple, but it's, again, sometimes difficult. So central complex is a conserved brain region in insects. If you see grasshoppers in your backyard or beetles at your home, cockroaches, they all have the central brain region. That's what makes insects intelligent. It has 4 structures. One is like mustache. You see this bar shaped. It's important for locomotion. And the other one is fan-shaped body in grasshoppers, for example. Here in flies will be more sophisticated. And then there's a donut. It's called the ring. It's like a circle, but in grasshoppers is a half circle. In flies will be full circle. It's called donut, ellipsoid body. It's important for navigation. And you will appreciate today we have found some neurons which project to the fan-shaped body in flies, and they are important for sleep. And many of the new studies have been implicated or have been involved with for understanding what are the function of other neuropil regions like moduli we don't know much about, and also fan-shaped body. Most of the neuron types, it has more complexity of different classes of neurons, but we don't know yet how many behaviors it is associated with. So insects are a good model system to understand higher order brain structure, formation, and function. I think you'll appreciate, like I do this experiment, very simple, from lung, like in Sweden, they study migration. This is a dung beetle rolling, like I mentioned earlier. It will go in the opposite orientation and go fast towards its nest. But now when you change the sun by putting shadow and effective sun, it will get confused. And that's how fast the integration of all this information occurs in an insect brain. Then it goes back on the other side because it feels the home is the other way. And now again, it goes back to the other side. So that tells us how integrate this brain region is to make these higher order brain decisions very quickly. So if you This is my second PhD student. So she drew this as an abstract art of what we do. So we are trying to understand our own brain diversity, different kinds of neurons, how they are formed using fruit fly brain. That's what I mentioned in my talk. The title is Tiny Brain, but it has huge impact. And also this guy appreciates this. Actually, this is when I was interviewing at UNM, and I had this baby, first one. He also likes flies. So now back to flies. So we will focus now on flies. In flies, there are two phases of neurogenesis, means formation of these neurons. One is embryonic, means early on, when the flies lay eggs. These stem cells, which are called neural stem cells, will make neurons for the maggot. Maggots crawl. They do some interesting behaviors, but they don't fly. Not as complex as the adult fly. At the end of embryonic stage, these stem cells will not divide anymore. They will go quiescent. When the maggot hatches, or when the maggot comes out of this hatched, fertilized embryo, they will again start dividing. And in maggot, in flies, is 5-day long period. And then at the end of the 5 days, they will make a cocoon, like in butterflies, and it'll be called pupa. Lots of cell death or metamorphosis happens here. The brain is transformed to make the adult fly. Many of the neurons which are in the adult are born, mostly are made during the larval phase. That's why it's interesting, because we try to figure out how do these stem cells in the maggot brain make different classes of neurons for the adult flies. As I mentioned earlier, there is a donut-shaped fly called ellipsoid body. This would be ellipsoid body. This is a figure or picture from Genelia, which is a big neuroscience campus, and from our collaborator, Vivek Jayaraman. He has shown that this brain region is important for insect navigation or fly navigation. And you can see a fly doing interesting behaviors. For example, here, Fight Club. How many of you have seen fruit fly fight? This is Fruit Fly Fight Club. And on the right is a movie, real-time movie from my collaborator, Vivek Jayaraman, and the fly is on a ball moving. And you see the ellipsoid body. These neurons will be active when you see bump of activity, like more bright colors. This is live fly on a ball. And you can see then on the left or the right, when the fly tries to turn left or right, you can see the neural activity happening in those brain cells. And with the sophisticated technology we have in fly, that makes it a good system to study. So fruit fly also is a good system because it's only 15 days. We don't have to wait too long. Male and female mate, and then lay embryo. And this is the larvae, 5-day, as I mentioned. It will be L1, L2, L3, larval phases. And then pupa is 3 days, and then you have an adult fly. So you can study development also, and also function of the adult brain in only maybe a month. And what's also interesting in the genetic screens, so you can do genetic screens and try to identify or identify what are the genetic mechanisms regulating brain development. Like this is a screen which was done in my postdoc mentor lab. This is the wild type brain, maggot brain. And if there is something wrong during division, you end up with small brain. This will be less neural stem cells. And sometimes you get more of stem cells. Stem cells keep on dividing. And if there is uncontrolled division, you end up with brain tumor. And 5 years later, same gene does similar thing in humans. It's called BRAD, brain tumor. Anyway, so it's a good system to understand developmental mechanisms and neuroscience. And also genes are conserved from flies to humans. 70% of genes are similar. So again, fly power, you see, we have central brain here. Insects, maggots have central brain, and then will be ventral nocord, and then the neurons, which go to the peripheral systems. And adult fly also has central brain. So we'll be focusing mostly on the central brain today. Also, other important discoveries have been made in flies. For example, formation of eye. There's a gene called PAX6 or eyeless. If you have eyeless in this place, you make up eye. If you don't have eye. Same in humans. If you put eyeless, it's a good, usually I show in my class. So if you put eyeless on the legs now of a fly, you get eyes on the fly leg. Anyway, so Halloween stuff. So we can understand genetics of it. So now going to the maggot, this will be a maggot in a light microscope. If you come to our lab, I can show you like this. And then if you do more confocal imaging, which is a big microscope, we have this will be central brain, and this will be ventral nerve cord. And then these will be different classes of neurons which go to the body segments of larvae. And larvae crawl, you can even image muscles. This is not what we do, but you can also image muscles. And also, you see, this is called, see this fly on the right, you activate one neuron by heat. The flies go only backwards. So it's called Michael Jackson or moonwalker neuron. So one neuron is activated by putting it on higher temperature, and you end up making fly go in the opposite direction. And this we discussed, they fight. The other thing which is cool in insects or also in mice is we can remote control their behavior. For example, this is a maggot, and we have put channel rhodopsin in their neurons. And if we put light on, the neurons will be active. You will see in control, nothing happens. But after you see when the neuron has channel rhodopsin, the larvae will freeze. So in that way, you can dissect circuits in maggots or in flies, which neuron is important for which behavior. And also what is cool is you can see what's happening in the real time in a fly brain. For example, this movie is from New York Times. This is a fly brain.
How do you watch a fly's brain in action without putting it on a leash so it doesn't fly away? Answer. First, the fly surgeon removes the top of the fly's head. Then he glues on a tiny see-through panel. The fly seems okay, but it gets to rest for a day just to be sure. With a sophisticated 3-camera setup, the researchers can track the fly while seeing into its brain, like courtship. But this male fly, with its visible brain, is courting a female. The fly was genetically engineered so that any neurons that are active light up when hit by a blue light laser. Fly brains can tell us a lot about all kinds of brains, including our own. So
you can see lots of interesting things happening. This we discussed early in a real time. And I don't know if you want to see this, but since it's a Halloween month, we can show it. So these will be flies where one neuron in the thorax, like neck, has been activated by heat. They don't have head, but you will see what happens. They will fly. So they're called headless fly. So that is a sophistication of fruit fly genetics. And I don't know if you want to see fly grooming. You can see flies clean their face and their eyes. There is a stereotypic coordination. You first do one behavior, then you go to the second step of the behavior, then you at the end you clean their eyes. So anyway, there are circuits, there are neurons behind all these very interesting behaviors. I think this we can skip. This is again power of a fly. So we can also study disease models like epilepsy. For example, here, this lab has control flies and also epileptic flies where gene is mutated. And then if you shake the flies a little, and you can see the normal flies will recover, they will go up, while as epileptic flies will have seizure, and they can, this usually are at the bottom. And you can see the seizures happening in their twitches and other things. Now this is done. So now going to our own research program. What do we do? So as I mentioned, we understand how these different classes of neurons, which you see saw lots of different kinds of behaviors a fly does. That behavior requires different classes of neurons, like we in our brain. So how do these classes of neurons arise during development? Now we go to stem cell. This will be maggot brain, not a real image, microscopic image, but a cartoon. So there are two classes of stem cells, type 1 and type 2. The red ones are type 1. They will self-renew and make 2 neurons in each division. And over 5 days, they will make 100 neurons. But we work on these, what's called blue cells. They are called type 2s. You don't have to remember the name, just remember that they will not only self-renew themselves, but also their kids or the progeny are also stem cells like this guy. It's called INPs. In a way, they amplify the neural pool. Each type 2 stem cell makes over 400 neurons or glia. And this is the real image, microscopic image of these. Green will be all these blue stem cells, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, and DLs2 on the other side are 2, on this side are 2. So we can go to these stem cells, and also we can track these stem cells from development to adult, which is not possible in mice, which is not possible in any other species of vertebrates. So there is a map. We know this stem cell makes these neurons, which is in green, which go to the central complex, bridge, fan-shaped body, and donut ellipsoid body. So each stem cell has unique contribution. You see, they don't overlap. That means each stem cell makes a distinct classes of neurons. And it's a good model system because it tells us not only which stem cell makes which, the lineages are also conserved, means the stem cells. For example, on the right is the fly, like I showed earlier. And these will be 4 stem cell lineages. On the left is a grasshopper, and they also have similar lineages. So from insects to humans, this is a good model to understand, because you see in humans, we have outer radial glia, ORGs, which make the INPs, which make our cortex more complex and more thick in terms of number of neurons. Similarly, they divide, the type 2s divide to make INPs, and our own ORGs also divide to make INPs. So there's also similarity in that. So overall, our goal is to understand how this maggot stem cell or neural stem cell over time will generate distinct classes of neurons of a fly brain. And also this was recently in news. So fly is the only complex animal where we have mapped, it's from Genelia again, Genelia Research Campus, it's HHMI campus. So they have lots of money, and with Google, they have made the fly brain map. So each neuron we know is connected to which neuron. It's a big 10-year-long project. It's ended now. So now we have each neuron, what kind it is in the adult fly brain. We know where it is projected to, where it's connected with. And such fit will now be possible maybe in next few years, or maybe 10 years to in a mice brain. So we understand now how these stem cells generate the complexity of the central complex. So this was my postdoc. This is the only postdoc slide. I showed that these stem cells in maggot brain will divide, and they express lots of interesting genes. All these genes are conserved in humans. And later on, these stem cells will express receptor for hormone, like we have thyroid hormone or growth hormone. Insects have ecdysone, which is important for many things, but here also important for the transition of these genes in stem cells. So what do these genes do? This is what we are trying to understand here. So what we are kind of hypothesizing, what we are trying to also study or investigate is that these factors, genes which are expressed in young stem cells, and also genes which are expressed in late stem cells or old stem cells, will make different classes of neurons. And the current circuits we study are sleep-wake circuit and olfactory navigation circuit. But today, I guess I'll have only time to discuss the sleep-wake circuit. So again, this is how does a stem cell over time make distinct classes of these neurons. So one neuron happens to be the sleep-promoting neuron, which is called DFB, dorsal fan-shaped body neuron. And this is the first PhD student. He has done amazing and lots of work. He's also from Kashmir. So this will be the real image of a fly brain, and this is the sleep-wake circuit. Green neurons are sleep-promoting neurons. They tell fly sleep when fly needs sleep. And also helican cells. Helican cells are like a circle here, and they will be inhibited. And this is a neural mechanism of sleep, and also there is circadian, which most of us know. When it is dark, we sleep. When it is light, we wake up. But if we don't sleep for a night or 2 nights or 3 nights, next day we are forced to sleep in the day. And this is the role of these neurons. They are sleep promoting. They tell our body to go to sleep when we need sleep. They're called homeostat. And for some of you, maybe many of you know that Nobel Prize for sleep was actually given to 3 people who work on flies. They showed that in flies, there are these genes, which are clock genes. They are active when there is light, and they are downregulated when there is light, and they are active when there is night. And similarly, in us, we have clock genes, and they also do similar things. So it's a good system. Again, the same kid when I started here is now helping us in our lab with fruit flies. Again, the time is flying. Anyway, so this is not published yet. We are trying to communicate in maybe a week or so. It takes time in our case also, even though flies are so fast, but it takes time. So this example is these sleep promoting neurons. The genetics is complicated. I don't want to talk about that, but if you see color, that means they come from these stem cells. So this is a maggot brain. This is the adult brain. In magenta will be neuropil, all neural structures. And in green are our neurons, which are sleep promoting neurons. And then we can also find which stem cell. It's called lineage tracing. Like we have 23andMe, we can track our lineage back to, I don't know, Europe or Middle East. So same in here, you can track lineages in adult. So you see in green are all more than maybe 100 neurons. They all come from this single stem cell. And if you see green neurons in there, that means they also come from the same stem cell. So many of them come from DL1, which is this stem cell, but there is one neuron, which is an outlier out of this cluster, which comes from DM1. You see this pattern is different. So we have linked sleep neurons to single stem cell. And then, as I mentioned earlier, these stem cells over time express different classes of genes. And in adults also, some of these genes are retained. For example, E93 here is ecdysone hormone-induced gene. And it was interesting to see that these neurons also express E93 in the adult. That led us to a hypothesis that maybe they are born late in development, means somewhere here in larval stages. And again, we have complicated genetics, but very cool. You can use that genetic trick and do heat shock. It's like dye filling. If you dye fill here, turn out switch here or here. If neurons are born after, you can see them green. If they are born here and you heat shock here, you will not see them labeled. And this is what Adil did. You heat shock these three important time points. You have neurons if you do here labeled. You have neurons if you heat shock here labeled. But if you do at 72, you don't see them. That means they are born somewhere here between 48 and 72 hours after larva hatch in the fly. And we know many of the genes that are expressed here. One of them, interesting one, was hormone receptor. You see ecdysone receptor. Similar in us is thyroid hormone. So we hypothesized that in stem cells, maybe this hormone signaling is important for formation of these neurons. And now this is interesting data. We have manipulated stem cells during development in maggot brain, but we are assaying the adult fly brain. So magenta will be neuropil and green will be DFB. So this is control for all experiments. We always have to have control. Neurons are normal. So when we knock out a receptor, it is called ECR, ecdysone receptor. We knock out. So there is no hormone receptor in the stem cell during development. You see there are no neurons formed. What does that tell us? That tells us during development, when stem cells are dividing, they need hormone signaling for the formation of sleep circuiting fly, which is new. Nobody has shown this before. And if the neurons survive, sometimes the genetic phenotype is not 100% penetrant. Some neurons survive, but they are lost in identity. They don't know where to go. They are supposed to go to this layer where they are regulating sleep, but they are everywhere. So that means ECR is important in late stem cell to form the adult fruit fly sleep neurons. This is kind of an interesting because hormones have also a big role in our own human brain development. Okay, so how does this ECR regulate the formation of these neurons? So we knocked out the receptor, but when we knock out receptor, we also lose many of these genes, SYNCRIP, BROAD, E93. Is it because of this, or this? So I'm not showing you all the results, only E93, because it is through E93, how this phenomena occurs. So this is, again, we manipulate genes in the stem cells in larvae and then assay the adult flies. This is control, and if you knock out E93, which is hormone induced, so it's ECR and then E93, in stem cells, you lose these neurons. If you put more of them, you don't make more of them. So it's not sufficient. It's only necessary for the formation of these neurons. Again, then this was 2020 COVID. This is the second kid. And when you have kids, young ones, you lose sleep. So I got interested in understanding sleep now. If we knock out hormones in stem cells, do they have sleep defect? And I'm not a behavior person. We tried we have a good collaborators. We tried to do something here, but mostly for fun. So the results we are showing you today is from collaborators. So in flies, how do you measure sleep? So there's a tube, there's infrared beam. And if you put a fly in there, and if fly does not cross the beam for more than 5 minutes, it's called sleep. It was the old characterization of sleep. And Nobel Prize was won on this small, simple instrument. But now we have sophisticated 3D printed tools where Galistro Lab has developed. We can have each fly in a tube and have a Raspberry Pi camera. And you can have minute, more information than just fly sitting on one side. And Krishna was an undergrad when I joined, and he established this ethoscope in our lab. So we got 3D printer, and he did some recordings. But we wanted to get more confirmation from people who are experts. We reached out to Ori and Buddha. They are at CUNY, New York. So what was interesting is control. This is not control. This will be the mutant. They didn't have overall sleep defect pattern in daytime, which is flies sleep also after lunch, called siesta, like we do sometimes in summer, and also mostly during nighttime. So there was no significant difference in overall sleep between daytime and also nighttime, between control and RNAi. But what was interesting is this is a new concept in the field that, like us, we have shallow sleep and deeper sleep. So when we calculated deeper sleep, which is more than 60 minutes of inactivity, this was significantly reduced in flies where we knock out receptor or hormone during development, both in daytime as well as in nighttime. So what does that mean? That means these sleep-promoting neurons are important for sleep consolidation, means that forming deep bouts of sleep, which is in us will be REM sleep. In flies is more than 60 minutes of inactivity. Although overall sleep in our mutants is not affected, but long bout sleep is defective, means they sleep in small fragments, like fragmented sleep, which is also interesting because many of the autism or neuropsychiatric disorders or ASD patients have fragmented sleep. And again, we reached out to our collaborator, Matt Kaiser. He is a neuropsychiatrist, studies sleep patients in his clinic, but uses fruit flies to understand the biology in his lab. He has shown that, like in us, in humans, there is infant sleep or juvenile sleep, like kids need more sleep, at least until 5 years, because it's very important for the maturation of the nervous system or circuits. Same thing he showed in flies. The fly juvenile or a 3-day-old, when they hatch, sleep more, which is important for the maturation of their circuits. So he also wanted to know if, because these neurons are important for that, also deep sleep in juvenile flies. So he wanted to see if there is any difference compared to two controls. You can see here, this is our RNAi, our mutant. There is a huge difference. The sleep bout number increases. That means both in day and night. That means the sleep bout, which are 60 minutes, is fragmented into small bouts. That's why bout number increased. The bout length decreases. So that means both in day and night time. That means, again, sleep is fragmented in the flies where sleep neurons have been absent because of hormone not being present during development. Now you'll appreciate how important it is to have hormones at a particular time in stem cells to form these circuits or sleep neurons. And if something goes wrong, either in the signaling or in the environmental mechanisms, you can see here flies on the right side, even in the day, night one are more active. They don't sleep properly. So I think I don't have time to go to the olfactory navigation. This is the second project, but I would want to highlight a little bit about the diversity, if there are time, a few minutes, 5 minutes or 3. Is that okay?
Yes, please do.
Because I do lots of, I guess, equally, maybe more even, mentoring undergrads and high school kids, I wanted to make sure where your support is utilized in an impactful manner or impactful way. So as you may have heard now, I founded JK Scientist when I was a PhD student in Germany. Now it is a self-sustained nonprofit organization, and the first generation I mentored, they are taking it over. Thousands of students are there and hundreds get mentored. And something I started here also when I moved, although during pandemic, but I was able to make connections with the Pablos of Zia and James. And also at UNM through NSF career, I have this education component where I teach every spring 10 undergrads. And the first cohort was in 2021. And what we train them is to become scientists. So they have never experienced research, then they do fly research for a month, and then they go to different labs or continue degree. And thanks to OPR, they gave us these 10 microscopes at the beginning of the course. And also what I realized is in my career, at least through my experience, it's very important for our students to show scientists from diverse backgrounds, coming from similar backgrounds. So I invite also scientists to UNM or on Zoom to interact with my class. For example, here is Andrea Gomez. She was here in 2022 class, and we are having lunch, informal pizza lunch. And she's the only Native American neuroscience, actually from Laguna Pueblo, I know of. She's at UC Berkeley. She was with us, and many got inspired. For example, Ethan in my lab is from Navajo. He got interested and excited and continued in my lab research. Gonzalo was an undergrad in my lab. Now he has started his PhD in Oregon. And there are many other students, like Alexa is also in my lab. And Carolina is my informal mentee. So I have been mentoring many students, and I have been inviting many scientists here, maybe more than 25 so far. It started with like introductory fall, which was usually in through Zoom during COVID, but then many have come in person, and many also continue to do in Zoom sessions. So all these are very famous international scientists, neuroscientists. And with my friend, Matt Clark, he comes here every spring, and we organize this Pueblo Brain Science Neuroscience Symposium. The first was last year, which was funded by Grass Foundation, thanks to them, and also NSF. So we have instructors, which are also from our lab, from undergrads, so that our undergrads know that they can be also grad students or postdocs or researchers. So we use mostly fruit flies for 3 days. And in these 3 days, we have high school teachers, high school kids, and undergrads, and we train them to do science, or at least to get excited about science. And this year, we had also the second one. We had invited more guests from outside, more postdocs. And also, this time, the community was open. Zia and James, Zia Pueblo students were present in our workshop this year. So it happens every spring. This year, we didn't have funds, but I managed to get at least food from people who I know when I go to get some lunch with them, from them. And thanks to Yasmina Cafe and Bawarchi and Starbucks for free coffee. It's very important to sponsor these activities. And these are some snapshots and some pictures. You can see many on my webpage. And this is Matt Clark, who comes here every spring from Pennsylvania. And these are my PhD students, and these are teachers here. And I am part of this Pueblo, and Zia and James, they invite me to their education retreats. Like this is my grad student. We show their educators and community. This Krishna, he uses PIVR at 3D printed tools to as a fly insect behaviors. And we also go to their schools. And this is one example. We go to many schools. And they invite us to their youth festival. This was last year, and we were there for 2 days, and also, again, using fruit flies to excite about science. This is, again, finding your roots. What are my roots? Kashmir, this is what I was mentioning earlier. I started it. Now it is with the safe hands of these all scientists at different places in the world, and they are taking it forward so that I focus on New Mexico. And these are all the amazing people who have been doing all the science. Collaborators, Kathy, very helpful and very productive collaboration. We are almost accepted at current biology. The first paper, which I didn't discuss about olfactory navigation. Matt Kaiser is asleep. And all this Buddha is also sleep. And Matt, he comes here for the workshop. And my mentors, I think mentors is who have made me who I am. Mentors shape lives, and I would take this opportunity to appreciate their contribution. And in Flyfield, we share resources, and I thank Pablos of Sandia and James. And NSF, this all has happened because of this initial boost I got, NSF Career Award, and I didn't know how to even write a budget. And again, OSP helped me a lot, and they still help me in making budget justification. And all these are very important because that way we can submit more and more grants. And Alfred Solon recently, this was kind of surprise. Usually they don't fund any in places like New Mexico, but again, somehow maybe somebody was there and got my idea of how stem cells can be useful or flies can be useful to understand neuroscience. This is very good award to have. And recently, NARSID award, which is also very good in neuroscience. And also, I would like to thank you all for helping us in keeping our research program funded. And thank you. This is the kid who was born in 2020 now is looking at fruit flies. And this is why I send these fruit flies sometimes to also kindergarten. And this is their reaction you see here.
No, it is not.
Anyway, so this is what I wanted to share today. I don't know. I hope it was on time.
Thank you. Bye.
Yeah, that was great. Thank you so much, Dr. Syed. I appreciate it, and that was an excellent learning opportunity for everyone. We are at time, and I apologize because we did get a late start. But Dr. Syed, if anybody has questions, is it okay if they reach out to you via email just to learn a little bit more about your research, or if
yeah,
they
thank
just
you.
have questions
Please
about
feel
what's
free to
going on?
reach out to me, and if you have a high school kid or neighbor, they can always come and get their hands dirty in our lab. And if you see a fruit fly next time in your kitchen, do not kill it. Send it here.
Sounds great. Well, thank you again, Dr. Syed. That was a great talk. Appreciate the information, and as always, I thank you for taking the time out of your busy day and busy schedule to prepare this presentation and tell us research administrators a little bit about your work, and also how we, as research administrators, we don't always see how we influence or contribute to the work that is done by researchers at the university. So I appreciate you taking the time to tell us a little bit about your research and how we contribute to that as well.
Thank you very much. Thank you. Bye.
Right. Okay. Thank you for the others who are attending, and we have our breakout session scheduled for 10:15. So we do have a break for 14 minutes. So if we'll join back here in about 10:13, and then at 10:15 we will do some work on sending people to their breakout session. So see you very soon. Hello, everyone. I understand some of you might not be at your computer because we're on a break, but we're actually having some difficulties with this Zoom link and breakout sessions. And so unfortunately, we're going to have to switch Zoom sessions. So I'm going to put the updated link in the chat. We'll also be sending out an email to everybody who is registered. And we will, as soon as, we can start joining this one as soon as we'd like, as soon as you're able to, and we'll still be able to send everybody to their proper breakout sessions at 10:15, but we will have to join a different Zoom session. So I just put that link in the chat. Marissa should be sending out an email to everybody who's registered. You'll also have it there, and we will see you very soon in the new Zoom. I'm going to stay here for a bit for people who maybe were at break and not at their computers, just to let them know, but see you in the new breakout or the new Zoom session very soon. Thank you, and apologies for the confusion.